you go live.
Yeah.
So just a question, Dr. Sharma, how much time you would require to take the audience to the presentation, like how many minutes?
So means I would speak maybe probably for 15-20 minutes, and then
All right.
then we can just take questions.
Sure, sure.
So, ma'am, it's 3, 2,
Mhm.
1. We are live to the audience. We can start this program. Over to you, Dr. Varsha, ma'am.
Thank you, Prayag. A very good evening, everyone. I'm Dr. Varsha Pawar from Lupin's medical team. Lupin is a transnational research-based biopharmaceutical company with a global presence. And as a company, Lupin recognizes the importance of scientific knowledge exchange. And we always endeavor to facilitate propagation of scientific information that will be useful from clinical practice perspective. It's really an honor and privilege to have you all doctors with us for today's session on the very interesting topic of myths and truths related to biosimilars for retinal disorders, and for which we have a very eminent and honorable speaker with us, Dr. Ashish Sharma. Dr. Sharma is considered as a world leader in the field of biosimilars for retinal disorders. His work in this field is recognized worldwide. He is the lead author of 20 indexed publications providing key insights about biosimilars in the field of retina. He has been an invited speaker by various European and American societies of ophthalmology on this same subject. Dr. Sharma will be speaking about his experience on biosimilars for another 15 to 20 minutes, and followed by which we'll take up the questions from the audience. So I request audience, you can start typing your questions in the chat box. Uh, uh, as of now, also you can start, no issues. And a very warm welcome to you, Dr. Sharma. Request you to take over the session from here onwards. Thank you.
Okay, thank you so much, Varsha, for that very kind introduction. And Miss, I would definitely like to say thanks to all the audience, Miss, whosoever is connected today. It's a weekday, and it's a summer holidays for children. So thank you so much, whosoever is connected. So I would briefly be talking for maybe some 15 to 20 minutes. And then probably we would just shift to the question and answer sessions if anybody has any. So feel free to type your questions. This interaction is always, that's what I like. So I will share my screen. I hope my screen is visible.
Yes, sir. It is visible, sir. And
okay, now the full screen.
yeah, yes, sir.
Okay. So I'm going to talk about, that myths and truths. Why the topic was chosen is that, I've been speaking on various platforms, both national and international, since last 2 to 3 years about biosimilars. So what I found that it's not only India. India is far mature than Western world at this point, if you talk about biosimilars. So, but still, you have newer biosimilars coming in, then people have different kind of perception, especially in terms of safety. So let us just talk about and understand whether there is a, what is the real truth and what are the myths, and whether we can bust some myths during this next 15 to 20 minutes. Before I proceed further, these are my disclosures. So I do have disclosure with Lupin, which is sponsoring this meeting. So as I mentioned before, our group has been working and trying to understand biosimilars since last 4 to 5 years. We tried to find answers related to immunogenicity, safety, efficacy, switches, and not only that was for Indian molecules, but it was for all the global molecules, whichever was in pipeline or approved as far as biosimilars are concerned. So at this point, I would definitely like to highlight and acknowledge the group which has helped us in understanding these molecules across the globe. These are all our colleagues and friends from Europe, US, and my students. So a special highlight to them, special acknowledgement to them. So biosimilar as molecules, I'm sure whoever is attending this webinar, you must have heard the name biosimilar. I would just like to mention that biosimilar are molecules which can be produced by a company whenever an innovator molecule patent expires. For example, Lucentis is the molecule for which biosimilars are available nowadays. India was a country which could bring Lucentis biosimilar, at the earliest in 2050, because India was not part of a global treaty that time. That was the reason even Lucentis, patent was not expired, but it still we could bring biosimilar of ranibizumab, but that would not happen over the time with other biosimilars. So essentially, let us understand that what biosimilar molecules are. So biosimilar molecules are the same molecule, similar to an innovator molecule. Let us take an example of Lucentis. Lucentis has, a ranibizumab in it. So it is a ranibizumab molecule, but it is much, much cheaper because you do not need to spend a lot of money and investment in clinical trials, larger clinical trials, because you already have an innovator molecule which has been found very safe. But it's not something that these molecules are not tested. These are very well tested because regulatory has a specific guideline, both in India, overseas, FDA, European guidelines, and they want to make sure that these molecules are similar in terms of safety, potency, and purity and efficacy. So there should not be any clinical meaningful difference. Clinical meaningful difference, there should not be any clinical difference in safety, efficacy, purity, and potency. But they are not the same molecule. So the reason is because any biosimilar, even if you talk about a Lucentis coming from US, a Lucentis coming from Europe, there would be some differences. So it's not something it's about just biosimilars might have some minor differences. It could be even Lucentis might have minor differences. But the regulatory always makes sure that these differences are not clinically meaningful. So that is what the biosimilars are. Again, just to highlight, India has maximum number of biosimilar ranibizumab molecule at this point. No country across the globe has aflibercept molecule at this point because aflibercept patent has not yet expired. But a lot of molecules are in pipeline. Probably by 2025 we will have them. So as of now, you have biosimilar ranibizumab. And if you talk about India, you have three predominant manufacturers for biosimilar ranibizumab. One is Intas Pharmaceuticals, which was approved in 2015. The name of the molecule is ranibizumab. Then comes Lupin Limited. They have a molecule called Reniice. And then you have Reliance Industries. They have a molecule called Renizoral. So these are the three major companies. Now, sometimes you must have seen some representative coming to you with another name, Osiva. Osiva is nothing but the same molecule from Lupin, because as of now, Lupin is the only company which is producing it, not the Sun. Similarly, Cipla also, you might have seen some Cipla representative might be coming to you with some ranibizumab biosimilar, but it is not the production machinery. Production machinery is from Reliance. So essentially, three manufacturers. Now, it is not only India. If you look last couple of years, you have biosimilar ranibizumab approved globally. The test name of one molecule, SB11, which is approved with the name of BioViz in Europe and US. FYB201, another molecule which is approved with different names in different countries. In US, it is known as Cimerli, in UK, Ongavia, in Europe, Renibizumab. Japan also has ranibizumab biosimilar. So, the reason to highlight this slide is that it's just not the only India where we need cost-effective medications. There's the entire world which require cost-effective medications. And the reason is that's the reason that you have even more molecules in pipeline, 3 more molecules for ranibizumab which might come. Aflibercept, if you really see, that would be the big game changer across the globe, especially for India. Because as of now, aflibercept is a molecule which has been globally utilized with the maximal strength, with the maximum volume, but we do not have that volume just because of the price, because it's a costly molecule. So Cost does play an important role, and biosimilars do provide a big cost benefit, at least 40 to 60%. More biosimilars you have, more benefit, cost-effective benefit that you have. So you can actually provide patients adequate treatment. The reason that why cost is a significant factor is, if you really look at in your practice, my practice, it's difficult to give monthly injections. Hardly patients, they agree for more than 3 injections. So then our results are also compromised in that way. Avastin, which is still being used, but a lot of people, private practitioners, they do not prefer, especially in our country, because we do not have robust compounding pharmacy. So stability is a big issue. So biosimilars come with a boon because the pricing is almost similar that you pay for Avastin, and on a much, much safer and efficacious way. Now let us come back to bust some of the myth which requires clarification. Most of the time, people think, whenever they hear about biosimilar, these are not safe molecules. And this notion starts from what Intas had few inflammation with some of their batches in 2015-16. But you really need to think and see the overall picture now. India has 2 more biosimilars that I've already told you in the previous slides. Does any of the audience have any kind of inflammation associated with them? Most likely no. So what has happened over the time, and even if you look at the resume, after initial few batches, now you do not see any inflammation. So we really need to think about on this point rather than straight jumping on the safety issue. Now the question that I would ask is whether it was just Indian biosimilar. If you really look at Lucentis, when it was launched in 2006 and 9, their major trials, they showed inflammation of 17.1 and 20.9%, which was very, very high. But do you see that inflammation now? You do not. Any of the FDA and EMA approved biosimilars do not have inflammation, significant inflammation, which should be more than 1% now. So think about it rather than just jumping that these molecules are not safe. So I'll tell you the reason that why that has happened. So before 2015, the guidelines were followed were the ISO guidelines. And ISO guideline endotoxin limit. I'll take a minute to explain endotoxin. If you talk about biosimilars or any biologic, because these are medications which are produced in live cells, and you use a lot of buffers and other additive things. So those are the impurities that each and every biologic has. But impurities should not cross a limit. And these impurities are called endotoxin. Endotoxin is something which might cause inflammation. So previously, before 2015, the limit was 0.5 EU per ml, endotoxin unit per ml. That was good. But still, FDA realized that in spite of that limit, 0.5 EU, majority of the patients are okay, but still, some cases you do see some inflammation because of that limit. And then this limit was further strengthened, and companies were told that they should follow and they should restrict endotoxin limit up to less than 0.2 EU per ml. And that was a game changer. And since then, you do not see any inflammation in any of these molecules. So that's the reason. It's not just that molecule is unsafe, it's just because the limit was not tight. Now, second myth is biosimilars are not tested well in clinical trials. Now, if you look at the trial, Resumab was tested with 104 eyes, with Resumab arm 78 and Ranibizumab arm 26. Ranibizumab was tested in 160 eyes, with Ranibizumab in 107 and Ranibizumab in 53 eyes. So overall, if you really talk about the trial, phase 3 clinical trial, the most robust trial that you have is from Reni eyes. Means I'm not saying this because I'm speaking on Lupin platform, but it is true. As of now, whatever product that you have in India, as far as biosimilar Ranibizumab is concerned, Reni eyes phase 3 trial was the best designed phase 3 trial as of now. But now the question is, there is variability. There's variability in numbers. Will that reduce your confidence on the drug? And then you see the total study duration is 3 months, 6 months, and 2 months. And you see the primary outcome is approximately at 12 weeks, 16 weeks, 12 weeks, short endpoints. Will this, reduce your confidence on these drugs? Should not. So the reason is all these medications, the chances of inflammation is less than 1%. To really find less than 1% chance of inflammation, you really need to test. So, and the clinical number and the sample size should have been really, really large, which is not possible, which is not done by your FDA or European studies, etc. And there's a possibility in Europe. If you look at UK authority has already waived off phase 3 clinical trial for biosimilars. I'm not saying that is happening now, but UK has already accepted it because these drugs can be very well tested on the basis of their equivalence with the existing drug, which has already shown safety on a larger extent. So even a smaller trial is sufficient. So that should not take off your confidence. Now I'll come back to the end point. A lot of people, I was in Portugal last week, and then we had a debate on plus and minus that whether these drugs are great. So again a question came up that the trial is very short. Primary endpoints are very short. I'll tell you there is a reason for it. Look at your practice, your own practice. If you look at your practice, whenever you start anti-VEGF injections, what is the time when you have the highest effect and efficacy of that drug? You would agree with me that initial 2 to 3 injections. So that is the reason that whenever you compare, for example, Lucentis and a biosimilar, then you have to check their efficacy, neck to neck only at 8 to 12 weeks. That is the only way that you can check that how equivalent these drugs are. If you go 6 months, 12 months, you cannot check the bioequivalence. You cannot check the biosimilarity. That is the reason that these trial endpoints are short. This was the article which we wrote by the request from FDA reviewer, because he asked us that even in US, people do not believe that, that this is the truth. So it requires an understanding. So do not think that these trials have very short endpoints, so these are not strong. It is just because these are biosimilar drugs, they have to prove their similarity. That's the reason. But if you talk about efficacy and safety, that is monitoring throughout the study period. In US, 12 months, in India, approximately 6 months. Now, another myth which I see and which I hear from a lot of people, that biosimilars, they have done just one trial. And after just one trial on probably AMD, they got approval from RVO, they got approval for DMD, they got approval for RVO, etc. Why is that? This also should not take off your confidence on these drugs. Why is that? I'll tell you. If Lucentis would have shown any safety differentiation, I'm sure everybody must have used Lucentis, and you must have attended a lot of scientific sessions. Have you ever heard that Lucentis was much safer in DME compared to RVO, compared to AMD? Never. So if Lucentis has not shown any differentiation in terms of the indication, that's the reason that regulatory does not want separate trials. It will unnecessarily increase the cost of the drug, because the receptors are same here, VEGF. So this is again a myth that we all should understand. Now I'll come back to some of the examples, case examples that we can understand that how efficacious these drugs are. So if you really look at the case examples, means my experience has grown with RENI-Eyes in last few years very well. So because it's a Lupin webinar, means all the biosimilars are great, means I have used all of them. But because I'm speaking on behalf of Lupin, I share some of the examples that from my practice. So this is an example of a diabetic macular edema, case number 1. Patient had neurosensory detachment. After one injection, you can see there is some intraretinal fluid persisting, but neurosensitivity edema just responds really, really well after a month. Another case, diabetic macular edema, you can see large neurosensitive detachment, a lot of intraretinal fluid. And after one injection, approximately around 15 days, you see entire intraretinal fluid is resolved. And you see a lot of improvement approximately by 80% in neurosensitive detachment as well. And then this case was further treated. Patient wanted to have even further cheaper injection. We shifted because we have a chief minister scheme here which provides Avastin free of cost. So we shifted that patient back to Avastin. So patient Avastin was given, but Avastin did not do that great in this patient. And then we shifted, because Avastin was not doing well, shifted this patient on IVTA. In spite of IVTA, there was some fluid left, but this was not a follow-up for a month, so we would see patient is yet to follow up. This is another case of diabetic macular edema. You see, this is diabetic macular edema with 6-12 vision. After one injection, after a month, you see some reduction of diabetic macular edema. Patient visual acuity improved by 6/9. Some edema still persists. So whenever we have such situations, especially on 6/9 vision, it is not necessary that we aggressively treat these patients as per even the protocol B that from BRCR net. If patient has no complaint, patient vision is 6/9, observation can be done very, very well. So observation was done in this patient. This is case of a retinal vein occlusion. So we shift our indication here. So this was a fresh RVO case. You see patient 6/9, but distortion was seen. You see a lot of hemorrhages, intraretinal hemorrhages there, and soft exudates after one injection. Neurosensitive detachment flattened up, entire intraretinal fluid is gone, patient improved to 6/6. But after, again, 1 month, as you see in many cases, patient edema came back again, second retinoid injection was given, patient again responded very well with slight cystic changes remains. So this shows the efficacy of the drug, that how efficacious the drug is. Again, patient was shifted to Avastin again in the CM Chief Minister scheme, which provides free Avastin injection here. So we shift them whenever they require. So this patient is still on treatment. And now this is another case of retinal vein occlusion. If you see fresh retinal vein occlusion on top, after one injection, patient improved to 6/9 vision, and subtle cysts are there, hemorrhagic improvement, a lot of intraretinal hemorrhage resorption you can see. So you see the efficacy of the drug. Another, we shift indication here, neovascular AMD. Neovascular AMD in top slide, you can see a lot of hemorrhagic AMD here, a lot of hemorrhagic PED with intraretinal fluid, thick blood. But after one injection, you see on OCT great improvement on OCT vision, improved by counter finger 1 to counter finger 3. But you see, the resorption of a large PED, hemorrhagic PED, and the foveal contour coming back after one injection. So again, shows great efficacy of the drug. This was a parapapillary CNDM. You see activity just next to the disc, and then fluid collection adjacent to it. Again, after 2 monthly ranibizumab injection, patient is completely flattened, and patient achieved 6/6. So this shows the efficacy of this drug. This is a very interesting case. I'm not sure, still we were in dilemma of the diagnosis, but we have been treating this case with a lot of Avastin. Around 10 Avastin injections were given. Patient had fluid always, but after one injection, fluid gone. And especially if you really look at, slide on top, there was kind of a knuckle. I'm not sure whether it was a very large choroid vessel or what, but we presume that was. And after one injection, if you see, even that resorption has happened. So if you really talk about these molecules have shown their safety, now you see India has gained great experience. And if you really see the most experienced nation across the globe for biosimilars, that is Europe. Since 2006, it's been 18 to 19 years that none of the biosimilar molecule which was approved in Europe has been withdrawn or suspended for safety and efficacy reasons. So believe on these molecules and pass the benefit of these molecules, because unless we understand as a clinician, then we cannot pass this benefit to our patients. That means I can say myself that now I can manage my family members with continuous monthly injections with the help of biosimilars without any hesitation. My all VIP patients, they get biosimilars. Means it's not something that I do not use innovator, I use them as well. But if you really talk about safety, again, I will leave and finish this talk that novel assets might be more unpredictable. If you look at brolucizumab, even trials could not pick up the inflammation. But if you talk about biosimilar, they emulate a product whose safety and efficacy has already been very well established. So chances of any backfire or any kind of surprise is much, much lesser with these molecules. So with this, I will finish my presentation that many lives can be really better, especially in a country like us where people pay from their pocket. So pocket is important. And what biosimilars have done for this country is that after each and every biosimilar, innovative molecules have gone into pressure and they had to start some of the schemes to bring the pricing down. So it's not only biosimilar which is providing direct benefit, it is providing indirect benefit to the people or clinicians who want to use innovative molecule as well. So once again, I would like to share my sincere regards to all the people who are attending this on really, on a vacation time. Thank you so much, Ethan, for having me here. And we can have question and answers now. I will be happy to discuss that.
Thank you so much, Dr. Sharma, for such a lucid and brilliant presentation. We do have a couple of questions. Shall we start?
Yeah, sure.
All right. Yeah. So first question from Dr. Vishwas Virajdar. He is asking, my question is, biosimilar have problem related to safety. And further to it, he is adding, if biosimilar shows problem, then as a doctor, whom should we consult?
Okay, the name is Viraj, right?
Vishwas Virajdar.
Vishwas Virajdar.
Yeah.
Vishwas Virajdar means I would just ask a question at this point that whether you have any personal experience with any kind of inflammation with any biosimilar which you have used in last 2 to 3 or 4 years. I'm sure you would have not had. So this is a perception. I was explaining in my slide. This perception started from when Intas in 2015 and 16 had inflammation, mild anterior segment inflammation with initial few batches. And I explained the reason as well, that because that was the time when the limit of impurities were not very strict. But now the limit of impurities are very strict. So now you do not see that inflammation with any of the biosimilars. So do not worry about the safety part of biosimilar at this point. In case, because 1% chance is there with even innovator, means less than 1%, even innovator molecule and biosimilar molecule. So is it as similar as that? And anytime if you have that, as a clinician, you manage and you know how to manage. You just need to report it to the authorities and to the company, and you manage the way you manage routine inflammation cases. And fortunately, the best part is none of the case have any kind of posterior segment complications. These are all mild cases, but now you do not have that. So just take off that safety issue from your thought. That's what I would say.
All right. Yes. Moving to the next question from Dr.
I see a question from Dr. Rajiv Gandhi. Potency of all biosimilars equal according to me. Yeah. So Rajiv, yes, I mean it's potency of all biosimilars are equal according to me. Yes, because these are all compared against the Lucentis, which is an innovator molecule, and the only difference is India and overseas. There is a slight difference in the trial. Because India, all the molecules were compared based on the loss of 15 letters. But in comparison to the other European trials, they were compared gain of the letters. So that was the only difference. But to really answer your question simplistically, they are all efficacious. They are all efficacious really, really well. Because that is the only way they can get approval. Otherwise, they do not get approval if they show any kind of difference.
Yes.
I see a question from Akbar Salim. In case of established CNVM, which is more efficacious, ranibizumab or aflibercept? So this is the question, which is not related to biosimilar, but I am happy to answer. So as of now, if you really look at the data for CNVM, I'm not sure, what do you mean by established CNVM? You mean probably by an old CNVM or chronic CNVM. Aflibercept definitely has an edge over ranibizumab because it has one more receptor which it takes care. So overall, even the globally studies have shown that aflibercept is a little better drug comparison to the in comparison to the ranibizumab as far as CNVM, especially if it is a polypoidal disease. So that's what I would say. And I think I missed one question from Mahesh, Mahesh K. So Mahesh is asking that what's the long-term prospects of biosimilars in India? So Mahesh, I think you and me can be really proud being an Indian and being a biosimilar users, because India would be probably one of the first country to bring even aflibercept as well. And I think credit goes to Lupin. Lupin is already working very well on aflibercept front. And once aflibercept comes to this country in India, you would see a big change. You would see a really, really big change in our overall practice pattern. As of now, our practice is that we are trying to use maximum Avastin, and then because biosimilar is filling that gap rapidly, and as of now, biosimilar ranibizumab is the only one which is available at a cost-effective price. So once aflibercept comes, then our prospects of this country are bright. More means in both the ways. Patients would get a better care, better medication, and on a much cheaper price.
All right. So we have one more question from Dr. Tushar. He's asking, can I prefer NEIs in ROP or any other biosimilar works good in this condition?
So Tushar means, to be frank, means I'm not an ROP caregiver. So my experience in ROP is not there with any of the biosimilar, but you can very well use biosimilar NEIs in ROP because means what I know the data, the endotoxins in RENIIs are way lesser than if you compare even the European and US molecules. Means that's what I know. So you should not hesitate. And probably, I would say that if you use RENIIs in ROP, you just definitely collaborate with us. We would definitely try to publish that experience and do not worry about safety, because other biosimilars and Lucentis has already been proven quite safe in these cases.
The next question is from Dr. Nitan Shah. He's asking, high molecular weight impurities, does they matter a lot?
So what was her name?
Nitan Shah.
Yeah, Nitan Shah. So Nitan Shah basically, yes, means any impurity matters. But as I said, the regulatory does not allow impurities to go more than the acceptable limit. That is what the endotoxin limit I was talking about. So if you have more impurities, you have higher endotoxin, and that endotoxin limit you have to maintain, which is approximately less than 0.2 EU per ml. So impurities definitely matters, but regulatory definitely takes care of it, and companies, they comply to that regulatory. That's why they never allow impurities to go up.
Right. So Dr. Rajiv Gandhi wants to know which are the upcoming biosimilars in India other than ranibizumab.
So what was her name? Sorry.
Dr. Rajiv Gandhi.
Rajiv. So in India, as of now, means you have 3 manufacturers. As I said, one is RENIIs from Lupin, then Intas, Resumab, and then Reliance. I am not sure you have anybody else coming in for ranibizumab biosimilar. But aflibercept biosimilar, a lot of companies are trying to be into this. But I think Lupin has an edge because they already have their trials in place, phase 3 trial in place. So ranibizumab wise, I think we would have these 3 major molecules. What I've heard is that Sun, which is taking at present molecule from Lupin, it is not confirmed. I do not know, but they while in a casual talk, they were saying that they would have their separate manufacturing later. But as of now, you can consider these are the three molecules. But I think in next 2 years or 3 years, probably we would have aflibercept biosimilar. That would really we should look forward to it.
Right. Dr. Chaitra wants to know, should we directly advise ranibizumab instead of Avastin?
Yeah, I mean, so what all these biosimilars they have done is that they have, brought down the pricing to a level that now we can actually get away from Avastin properly. So that is much better for especially private practitioners, and even in my personal practice, even if you see all the protocols also, means although visual acuity is equivalent, if you look at the protocol T results, but still drying capacity wise, ranibizumab is definitely a better agent compared to the bevacizumab, which is Avastin. So you can definitely, if price is not a concern, which is not nowadays with the help of biosimilars, you can straight away start all your patients on ranibizumab for sure. I see one question from Dr. Seema from Delhi that do you have any personal experience with new biosimilars like RANI-Eyes, and how was your experience? So I'm not sure, Seema, whether you saw all those cases which I showed. Those were all the cases with RANI-Eyes. I showed brilliant effect in cases of diabetic macular edema, retinal vein occlusion, and CNVM, both CNVMs, means whether it's a central CNVM or parapapillary CNVs. My experience has been really good with RANI-Eyes with no safety issues.
Dr. Sachin Bodhale wants to understand whether there is any head-to-head comparison which has been done between RANI-Eyes, Razumab, and Vizumab.
Yeah, so again, I want to clarify one point here. So Vizumab is Cipla. Cipla is marketing it. Who is a manufacturer? Reliance. So ultimately, it's a Reliance molecule. So at this point, we do not have any data comparing head-to-head all these three molecules. But realistically means that's a good idea. Means we can definitely check it and we can try it. But I have my all doubts that you would find much difference there, because all these molecules were compared with innovator molecule, and all of them have shown equivalence. So because they have all compared with the same molecule, so the chances of differentiation are much less. But I would, I agree that to really answer this question, we can have one study, comparing all these three. But you need to have really all the, all the data and all the baseline parameters has to be really under control.
Absolutely, sure, sir. Dr. Anmol Naik wants to understand, Lupin has compared its molecule with Lucentis, but why not with Acentrix?
Yeah, so there is, so if you really want to understand difference between Lucentis and Accentrix, so Accentrix is basically a molecule which is only known as Accentrix in India. It is same molecule. It is same molecule, same ranibizumab, but because to bring down the India-centric pricing, the company had to go with a different name. That's what I have heard from the Novartis. So there is no difference in terms of molecule between Accentrix and Lucentis. Same drug, same. It's just a name change because of the India-centric policies. And whenever a biosimilar you talk about, you're talking about the global. And so globally, Lucentis is something which is known and available from the beginning. That's why comparison will always be with Lucentis for any biosimilar.
Right. Dr. Chaitan is asking, what are your views in coming years if many competition rises? Use of bevacizumab will be replaced by ranibizumab? In this case, which brand would you prefer?
Yeah, so means I'm sure that is already on the corner. Means biosimilars are replacing Avastin, especially for private practitioners. They have replaced it already. Institutions would replace it over the time, because as of now, a lot of these practitioners, they are charging around 10,000 rupees or 11,000 rupees for Avastin, which and the biosimilars it almost cost you around 6,000 to 7,000 rupees nowadays. So the replacement is definitely on the corner. So we would see less and less Avastin over the time. And what was the second question?
What would be your choice among the available options?
So means what I would say is, as of now, my experience has been great with both Resumab and Reniase. So means over the time, as soon as your as your experience grows, now Reniase has already completed around 15,000 injections. There is no complication, no inflammation. So it's just you whenever something new comes in, you always start slow. I mean, that's what I do. And so means my preference would be either of these two, but means I'm sure other molecules are also good, but my experience has not grown in that.
Right. We have one more question from Dr. Tushar. He's asking that Lupin employee has been promoting us the comparison between Raniase versus other Indian biosimilar, but batch taken for the other biosimilar was less. How will you handle it?
So I'm not sure these that number of batches would count here. Means it's the batch to batch comparison. I think if I know the comparison which was done, that Reniase compared with US batch, with European batch, and with the two other batches of the other biosimilar which are available here. So that is a comparison. So it does not matter whether you take one or two batches more or less, means which I'm sure it is not being done. But it's just comparison of primary structure, secondary structure, tertiary structure of the protein foldings, and then what is the level of impurities, what is the level of endotoxin between all these three. And whenever you take, because as I already said, all these biologics, they go through a lot of live organisms, so you find difference even between Lucentis, even between batch to batch Reniase, between batch to batch other drugs as well. So that is the reason that you take at least more than one batch, so that at least you deny that kind of differentiation. But means I do not think that that way it's they are trying to promote it. It's just that comparison between endotoxin, what is the way they have filtered it, the way others have filtered it. And on that way, means I know the data that I think Reniase had the least endotoxin, although means all the drugs had under the limit, whatever is required by the regulatory agency, but Reniase had minimum, I think if I remember correctly, in the advisory board meeting, it was 0.08, which is much less. The recommendation is less than 0.2. So it's a very safe drug.
Thank you, sir. Next question from Dr. Akbar Salim again. Doctor wants to know which stage of diabetic retinopathy is the ideal time to start ranibizumab protocol.
So Miss are you? So I think that's a very simple question to answer. If you have read about the protocol V from DRCR-net. So protocol V from DRCR-net, it says any patient who has vision better than 20/32 with the diabetic macular edema, you need not to treat this, because these patients, they do not have any complaint, and because diabetes is a multifactorial disease, and if you control other factors, then probably you can control that edema. So any patient who after 20/32 means if he declines vision by 10%, means I would simple mathematics, I say rule of 10. If they decline by 10 letters, that is approximately 2 lines, or if they decline by 10%, if the macular thickness increases. Means usually visual acuity is something which DRCR-net has made it as a priority for treatment. So any patient who goes down below 20/32, in India, Snellen's, if you talk about, it is approximately less than 6/9. If they go below less than 6/9, then definitely you treat.
Dr. Chaitra wants to know, how do we decide PRP versus biosimilar in advanced cases?
PRP versus biosimilars. Okay, so I think this is a kind of a different ball game altogether. So I would say the simple question I think is asking is whether to use anti-VEGF or PRP. So probably whenever there is a proliferation, means PRP is something which gives a long-term effect. Anti-VEGFs are a short-term effect givers. Their effect lasts for only 1 month. That is what protocol means recent DRCR protocol was trying to understand, that whether we can keep giving anti-VEGF injections in PDR cases, and if we compare them with observation and laser, then what happens? So they also came to a conclusion that it's not really worth keep giving anti-VEGF injection to halt a PDR, because that becomes really non-sustainable. So in PDR cases, always we should prefer panretinal photocoagulation until unless they have some kind of diabetic macular edema associated with it. If diabetic macular edema is associated with it, then definitely you add injections, because that would take care of diabetic macular edema. So central involved diabetic macular edema with proliferative diabetic retinopathy, if that is the case, you can combine both laser and injections, but not in just PDR cases.
Dr. Rajiv Gandhi wants to know, according to you, is the potency of all biosimilars equal?
Yeah, means that's what I answered even before as well, that because they were all compared with Lucentis. So based on the regulatory, their potency is same. But means somehow in some cases means I found means Reniase and Resumab, they're doing really well. But my experience, to be frank, is not that large that I can really compare and give you a recommendation between three. All three works. All three works.
So, Dr. Sharma, I think we have covered most of the questions that were asked by the audience here. And
so I'll just read, if anybody's left, we should not leave anybody. Please. So, Chatra, we answered. Okay, I think Sachin, we answered. Okay, this we answered. Of course, we answered. Right. Sachin, we answered. Okay, Rajiv, we answered. Okay, I think yes, we did. You are much wiser and much.
No. Actually, I have been keeping track on all the questions, right. So I am sure we have not missed any. Right. So I think then accordingly, I think we have the time restriction as well. So I really want to thank you so much for this insightful and really interesting session. I think many doctors have been benefited from this. Many questions, that means a lot of interest has been generated. So thank you so much, Dr. Sharma, for taking out your time on this evening out of your busy schedule. And thank you again.
One question came, we'll just answer that. Should we stop injecting eccentrics altogether? So okay. I would say no. Means it's not like that. Means you see the pricing, whatever fits into the patient's pocket. Means eccentrics is also a great medication if that is the innovative molecule. So it's not like then a biosimilar would replace eccentric. Every molecule has its own place. So, and when there is evidence of traction already and extensive fibrovascular proliferation, is it advisable to inject Resumab? So it's all about anti-VEGF. Whenever means this is a big question which has been answered by many, which is not answered by many trials as well. So especially I can say that although there is a possibility that there is a switch happens from hemorrhagic switch to fibrotic switch whenever you give an anti-VEGF. But you need to really see that overall what is happening in the surrounding retina. If anything is which is really close to the fovea, you can avoid it. In the same way, you can avoid anti-VEGF and probably do a laser, but again, much far from your fibrovascular proliferation. But it's not really proven, so you need to really see the overall picture of the retina. If you have angry vessels, active bleeding, and diabetic macular edema, you can probably give an anti-VEGF as well.
Right. So I think no more questions. Audience, is there any other question? When there is evidence of, no, this has been taken care of. I think this has been answered. All right. So, doctor, we can call it that we have answered all the questions now from the audience. And once again, thank you so much for taking out your time from your busy schedule and providing this feast of knowledge for all of us. Thank you.
Thank you, Lupin team, for having me, all of you. And thank you, audience, and you guys have a wonderful day.
Thank you. So, Prayag, we can
Thank you.
end the meeting here.
Yeah. Thank you, ma'am. Thank you, sir.
Yeah, sure.
So from here now, I'm ending this meeting. Have a great evening to all.
Thanks, Priya.
Bye.
Thank you, Dr. Ashish.
Thank you, everyone.
Thank you, everyone. Bye.